everyone, my name is Jessica Del Cruz. Today is January 23rd, 2019. It is week 3 lab, bacterial morphology and staining techniques. This is going to be exercise 2, differential stains. Today we're going to be preparing bacterial smears via gram stains using the pure samples from the broths that it was inoculated in my previous experiment. Okay, so what we're gonna do is I am gonna grab I have my broths here, and then I have my slides all ready to go and labeled. So I'm gonna do is I'm just gonna open this, and then I'm going to sterilize the lid. Okay, then I'm gonna grab one of the inoculating loops. Just gonna shake it, make sure it's dry. Okay. And I'm gonna reach in there for bacteria. Okay. Pull it out, make sure it doesn't touch the lid. And place it on here on my slide. Okay. Now, after that, I can dispose of this into the 10% bleach solution, and I'm going to sterilize it again. Okay, and I'm going to place the lid back on. Okay, I'm going to let that air dry. I'm going to grab my next broth sample. Do the same thing, sterilize the lid. Okay, grab another inoculating loop. Here, making sure that it's all dry. Okay, and then I'm gonna grab sample. Okay. And then place it on the slide, like so. Okay, cool. Now I'm gonna dispose of that. Sterilize again. Okay, and put the cap back on. And this is for the, now I'm doing the epidermitis. Okay. Okay. In there. Alright, and then I'm just going to dispose of that. Oh my goodness. Okay. Alright, and then now I'm going to heat fix it. Just making sure that they're all dry. Yep. So we heat fix it to ensure that the cells adhere to the slide. So I'm going to swipe it through the flame 5 times. Okay. It's going to be 1, 2, 3, 4, and 5. All right. So I did the equal eye. I'm gonna do the Saccharomyces cerevisiae. Okay, cerevisiae. Okay, so 1, 2, 3, 4, and 5. Okay. And then next is the epidermitis. Wait. Okay. Let's make sure that we got that there. Okay. Ready? 1, 2, 3, 4, and 5. Okay. Alright. Now that I've heat fixed all of these, we're going to wait for 10 minutes and come back and we are going to start staining them. Hi everyone, so now that I've let my slides sit for 10 minutes, we're going to start the gram staining process. Okay, so I have my first slide and I'm going to grab the crystal violet. I'm going to add 1 to 2 drops onto the cells. 2. Okay, I'm going to let it sit for 60 seconds. So with the crystal violet actually dyes all bacterial cells regardless of the peptidoglycan thickness. Almost there. Just a few more seconds. Okay, now we're going to wash it with tap water. I'm going to wash it till it's clear. Okay. Alright, now we're going to grab the second dye, which is the PVP iodine. And we're going to add to this 1 to 2 drops as well. Or 2 to 3 drops actually. 1, 2. Oh, sorry. Okay, we're gonna wait for 60 seconds for this. And so the iodine actually reacts with crystal violet, forming a complex that allows dyes to better adhere to the bacterial cells. Okay. Halfway there. Okay, and then we're just gonna wash it off. Okay. Alright, now we're gonna grab our decolorizer. And so for our decolorizer, we're gonna also add 2 to 3 drops, but this we're gonna actually wait for 30 seconds. And so for the decolorizer, it dehydrates the gram-positive cell walls. It traps in dyes, and the decolorizer washes dyes from the gram-negative bacteria. Okay. Get ready. Okay, and then we're gonna wash it off now. And for the last step of the gram staining process, we are going to use the saffron. And this one we're also going to put 2 to 3 drops. 1, 2. Okay. And we're going to wait for 30 seconds. So the saffron has no effect on gram-positive bacteria. Saffron actually causes gram-negative bacteria to become pink. Okay, so I'm going to wash that off. Okay, all right. All right, so now that it's all cleared out, what I'm going to do is I am going to blot dry this. So now from here, I'm just going to blot dry this and let this dry. And I'm going to continue on to doing the last 2 slides. All right.